All right, so this is going to be our review on protein synthesis. Got to start with a review of DNA. So I'm just going to draw a little bit of DNA here and review the parts. All right, so let's take a peek. This whole great big thing you know as a chromosome. This is a duplicated one. And it has two halves. This side and this side, both are known as sister chromatids, and of course they're identical because one side is a copy of the other. They're held together in the center by a centromere, and little sections of this chromosome, in other words, sections of DNA, are known as genes. And if you look, genes, they code for estrogen, insulin, growth hormone. They code for proteins. So genes are the instructions to make proteins. Proteins will make your traits. And if you look, both sides are identical. The gene for insulin on both sides, estrogen on both sides, and growth hormone on both sides. And let's zoom in now on this strandy stuff that makes the chromosome, which is DNA. One half is called the nucleic acid. They're in a spiral double helix staircase. So there are two nucleic acids held together by hydrogen bonds. This is what makes up DNA. And if you look up close, nucleic acids are made up of smaller building blocks called nucleotides. And there's roughly about 3 billion plus in the human genome. Genome meaning all the human DNA. Now let's take a look at what are the actual instructions in DNA. You guys know this. It's the order of the letters known as the nitrogenous bases, the A, T, C's, and G's. Now let's look at the amount of letters here. How many A's are there? 1, 2, and there's a third one, right? Because T always pairs with A. So there are 3 A's, and there has to be 3 T's. Always the same amount. So if 28% of a piece of DNA was T, 28% is going to be A, because they're always the same amount. So how much is C? Well, if you add 28 and 28, 56%. That leaves, in order to get to 100%, that leaves about 44% left. 44% of the DNA is C and G. C and G have to be the same amount, though. So 44 divided by 2 gives you 22% C and G. And just remember, C and G, they always pair up, so they always have to have the same amount. Let's take a look now a little closer at a chromosome and explore a gene. Here's a testosterone gene, here's an estrogen gene on a sister chromatid. We're going to explore, or take a look at actually, these genes up close. I'm going to draw out a testosterone gene, a section of DNA that codes for a protein, the protein testosterone. And I'm going to draw out a gene, section of DNA, or a section of a chromosome that codes for the protein estrogen. Question I want to ask you, how is one of these genes different from another? You know how the proteins are different. They have different shapes, which gives them different functions, but how are the genes, their instructions that make them different? Take a look at these. There should be some obvious differences. One, clearly should be, there are different order of letters in a testosterone gene and an estrogen gene. And clearly one gene is much smaller than the other. So there's a different amount of bases as well. Now we're going to take a look or examine how a chromosome is broken down into genes, or DNA, which is a chromosome, is broken down into genes, sections of a chromosome, Genes control proteins. Proteins control your traits or function in your body. So ultimately, genes are the instructions to make a protein, and proteins control your traits, how you look, how your body functions. Let's see how we go from a gene to a protein. Let's explore this a little further. So I'm just going to draw up a diagram here of a cell with a nucleus, got a chromosome, and we're going to draw the building blocks and organelles necessary to make a protein from a piece of DNA. All right, the stage is set. Let's examine and see what this thing is. A nucleus. Hope you got that one. This big thing right here, chromosome, we mentioned that before. And a section of this thing we mentioned is a gene. They control proteins, or the instructions to make proteins, like insulin, a hormone. Remember, proteins are hormones, enzymes, antibodies, and receptors. We said both of these things are identical, sister chromatids. That's why we drew the insulin gene on both sister chromatids and in the same location. And this little thing down here, floating around in the cytoplasm, is our ribosome. This is actually where proteins are synthesized. So it looks to be a dilemma. If proteins are made at the ribosome, and DNA holds genes, the instructions to make proteins, how do they get out? Because clearly this big chromosome cannot get out of here. Big question. How do the instructions in DNA get to the ribosome to make a protein? So in order for the instructions to get to the ribosome, you got to make a copy of a gene. Like you're stuck in the library, you need the information in the book. How do you get the information to your home to make your report? You copy the information down, you leave the library, can't leave with the whole book, so you just copy the information and leave with that. That's what happens here. A copy of the gene's made, called mRNA. You see that it's only one nucleic acid wide. It's able to squeeze out of there. Let's take a look and see how this copy of a gene, or how a copy of a gene called mRNA is made. DNA has to split open. The hydrogen bonds are broken, exposing the nitrogenous bases, the actual instructions in DNA. And then an enzyme will come by and it will start to build the mRNA strand one by one. Notice in RNA, all RNA, U is in place of T. So T takes a time out and is replaced with U. This one nucleic acid wide, one gene long piece of RNA called mRNA, messenger RNA, is able to leave the nucleus. So it's made in the nucleus, process called transcription, and it's able to leave the nucleus and ultimately go to the ribosome, which is located in the cytoplasm. So mRNA will make its way down. Ribosome will take it in. And it's going to be read 3 letters at a time. Those 3 letters are referred to as a codon because they code for a specific amino acid. The first codon, AUU, it's going to cause the amino acid A, B, C, D, or E to come over. Well, A pairs at U, A pairs at U, and U pairs at A. So the codon has 3 letters, and it has to have or match up with the corresponding letters on the tRNA, which will transfer over the amino acid. So if you're looking, the codon has to match up with the 3 letters on tRNA, known as the anticodon. That causes a certain amino acid to come over, and you're going to build a protein sequence of amino acids in a very specific order. Those amino acids will be held together by peptide bonds, ultimately creating a protein, which is also known as a polypeptide. Many peptide bonds. Do have to go back and talk about this. We said this little trident-looking thing that holds the amino acid, letter E, is called tRNA, for transfer RNA. This RNA transfers or transports the amino acid, the little circle with the letter E in it, transports it over to the actual ribosome. And it only brings over the amino acid if the 3 letters on the tRNA match or correspond to the 3 letters, the codon, on the mRNA. Therefore, these 3 letters are the opposite of the codon. They're called the anticodon. Notice GCG matches with CGC, and all these codons match to the anticodons. So tRNA brings over the amino acids to the ribosome. Once all the amino acids are together, why do they fold into a specific 3-dimensional shape to give a protein a very specific shape? Because there's 20 amino acids and they have very specific properties, such as being charged, positive or negative charged, having both a positive and negative, being polar, being nonpolar, hydrophobic, hating water, hydrophilic, loving water, could be acidic, basic. There's quite a few properties amino acids could be. So how does this cause the chain of amino acids to fold in a certain way? Let's see. Here's a chain of amino acids, a polypeptide. There's a positively charged amino acid and a negative one. Positive and negative love each other. They're going to go towards each other. So the chain of amino acids will bend, starting to develop a shape. There'll be a little bond there. Well, what if you took that negative charge and you moved it to a different amino acid, different order? So if you change the order of the amino acids, what's going to happen? You already know. It looks like the protein's folding differently. And you still have the positive and negative bond. And this little guy right here, maybe is hydrophobic, is hiding out in the interior. Remember, hydrophobic amino acids don't like water. They'll be located here in the interior of the protein. Because outside the protein, in the cytoplasm or blood or anywhere, there's a whole bunch of water. So hydrophobic amino acids will stay inside. The hydrophilic will stay outside, exposed to the water. So therefore, the order of the amino acids ultimately will determine the shape of the protein. And you know, the shape determines the function of the protein. All this is controlled by the genes and the DNA. So let's take a look and see how maybe your regents might ask this. It looks like this diagram of this chart has 3 species, and each species has a DNA sequence, an mRNA sequence with some missing, and it also has an amino acid sequence. You have to find the DNA sequence, make sure they're complete, find the mRNA sequence using the DNA sequence, and then find the amino acid sequence using the mRNA sequence. So DNA to RNA, or DNA to mRNA, happens in the nucleus. And mRNA to amino acid happens in cytoplasm. These first 2 amino acids are missing. We're going to use the mRNA GAG to see what the first amino acid is. So G, second letter is A, third letter is G, So we could pinpoint where that is on here, and we found that GLU is the first amino acid. Second mRNA codon is ACU. First letter, A. So we're looking in that row. Second letter, C, looking in that column. Third letter is U, ACU, stands for THR. Complete. Species B. There's a piece of DNA missing. We'll get to that. This piece of mRNA, easy. C pairs with G, A pairs with U, not T, and C with G. But what about this piece of DNA right here? You got to go backwards. So if the mRNA that was made is ACU, what must have the DNA sequence been? A will pair with U, G with C, and then T with A. Remember, don't forget it's DNA. And now the amino acid sequence, you're going to use the mRNA, UCU. UCU stands for SER. GAA. Let's pinpoint this guy. Stands for glutamate, GLU. Species B, we finished. Species C, all done. DNA versus RNA. How can we tell if something's RNA? Let's look at DNA and RNA and compare. Of course, you know DNA, chromosome made up of DNA. It's made up of two gigantic strands of nucleic acids. One, and then of course the other one on the other side. Held together by hydrogen bonds. Looks like we got a T in there, thymine. And a sugar in DNA called deoxyribose is also present, hence the word DNA. Also, you find many genes on a chromosome. Chromosome is synonymous, means the same thing as DNA. So there are many genes on a chromosome or many genes on a piece of DNA. A copy of that gene, we said, is mRNA. We also said this little fork-like thing called tRNA, which holds amino acids, is also known as RNA. So we have a piece of mRNA, this long single-stranded nucleic acid, and the tRNA, the little fork-like looking thing. How can we tell they're RNA? There's a U present called uracil. T takes a time out, is replaced with U, and it's only one nucleic acid wide, so it's narrow. And it's only, at most, one gene long. A copy of one gene is mRNA. Over here, tRNA. Over here is mRNA. And the R stands for... what does the R stand for? The ribose sugar. And don't forget, the two sugars make RNA and DNA different. Hey, it's me. Good luck on your test. Use this video to study. Hope it helped you out. I'll see you guys all soon. Stay tuned for the next video. Should be on reproduction.